Hello friends, good evening everyone. Just let me know if the audio and the video are clear. A show of hands. You can send me your hellos. Let's see if you can see the audio and video clearly. Just send me a hello, a hi, something to let me know that you are with us. Okay, is the audio clear? Is the video clear? Can you see the iPad's presentation? Can you hear my voice? Is my face visible? Great, great. Good evening, everyone. And we are starting with a new topic today. That is, we are going to be looking at the lipids. If time permits, we will also try to start the vitamins, but let's see how it plays out. Now, if you have seen some of my videos which are there on my page on the Facebook Biochemistry by Dr. Nilesh Chandra, there I have talked a little bit about the aims distribution of questions. Something similar we will find in the NEET and the next exam also. The from the lipids and that too from very specific topics, so it doesn't make a lot of sense to go into a lot of details of a lot of topics of the lipids. Rather, to get the best yield we focus on some of the very important topics. Two subtopics which are extremely important is number one, the classification. So we will see the classification in a bit of detail, and second we have to focus on the lipoproteins. These are two aspects that you must be very clear about, because they are most frequently asked: the classification of the lipids and the lipoproteins. These are two topics which are most frequently asked. So let's start with lipids. Okay, so lipids as a class have a big problem when we try to study them. When we try to study them as a class, they have a big problem, and the problem is no common chemical definition. We cannot arrive at a common chemical definition which will cover all the lipids. So this makes the study of lipids somewhat unorganized, because a large number of dissimilar molecules on the basis of chemical features are clubbed together under the lipids. So a lot of scientists have tried to classify the lipids. The most acceptable among those incomplete classifications is the classification provided by the scientist called Bloor. So we use the Bloor's classification. The classification which is used to study the lipids is the Bloor's classification. Very broadly speaking, the lipids are a group of molecules which contain the alcohols attached with carboxylic acids. In addition, they are soluble in organic solvents. In addition, they are soluble in organic solvents. So these two features are commonly used to identify the lipids. One part is chemical property, one part is the physical property. So together we try to talk about the lipids. Let's look at the classification of the lipids. Broadly, we classify them under three categories. Broadly, we classify them under three categories: the simple lipids, the complex lipids, and the third one are not exactly lipids, and they are precursors and derived molecules. So we still call them lipids. So precursors and the derived molecules, or the precursor and derived lipids. Let's look at them one by one. Let's look at the three subgroups: the simple, complex, and the precursor and derived lipids, one by one. So we'll talk about them one by one, starting with the simple lipids. Starting with the simple lipids. Garamana. Thank you. Okay. So simple lipids are esters of fatty acids with various alcohols. So these are the pure chemical form that we had mentioned, the chemical part of the definition that we had mentioned. They are fatty acids attached to alcohols via the ester link. So they are esters of fatty acid with the various alcohol. The name says they are the simple lipids. Now we subdivide them into two groups depending on the type of alcohol. First group are the fats. In fats, the alcohol is glycerol. How is glycerol different from other alcohols? It is different because it has the three hydroxyl groups. So it is equivalent to three alcohols. So the esters of fatty acid with glycerol are known as fats. Some of these fats are liquid at room temperature. They are liquid at room temperature. In that case, we call them oils. Oils are those fats which are liquid at room temperature. And then we have the waxes. In case of waxes, the alcohols are monohydric alcohols. The alcohols are monohydric alcohols. So this is the simple lipid. Simple lipids are having the common chemical definition of being esters of fatty acid with the various alcohols. They can be fats, they can be waxes. Some of the fats are oils. Okay. Now we go to the clinically very important group, and that is the complex lipids. Now we go to the clinically important group, the complex lipids. The complex lipids are esters of fatty acid, definitely with the alcohol and the alcohol and fatty acid. But in addition to alcohol and fatty acid, they contain additional groups. They contain an additional group. Depending on which additional group is present, we have the different names. For example, if the phosphate is present, we call it the phospholipid. If phosphate is present, we call it the phospholipids. If the carbohydrate are present, we call them the glycolipids. Then we can have molecules like the sulfolipids, the aminolipids. All of these can be present. The phospholipids are further subdivided depending on the type of alcohol into the glycerophospholipid. If glycerol is the alcohol, we call it the glycerophospholipid. If sphingosine is the alcohol, then we call it the sphingophospholipids. So phospholipids are further subdivided into the glycerophospholipids and the sphingophospholipids. Alright, so the phospholipids, the glycolipids, and the other complex lipids. One very important point to note is all glycolipids contain sphingosine as alcohol. All of them will contain sphingosine as alcohol. This means if I say sphingolipid, the sphingolipid actually includes two subgroups. It actually includes two subgroups. One is the sphingophospholipid and second is the sphingoglycolipids. So remember, the sphingoglycolipids or the glycosphingolipids as well as the sphingophospholipids, both of them are sphingolipids. Both of them are a type of sphingolipid. Both of them are a type of the sphingolipid. And then we come to the third category, and the precursor and the derived lipids. You can see large number of names are there: the precursor fatty acid, the precursor glycerol, the other alcohols, the fatty aldehydes, the ketone bodies, the hydrocarbons. Derivatives include the steroids, the lipid soluble vitamins, and the hormones. All of these are included in the derivatives. So the three categories: the simple lipids, the complex lipids, and the derived lipids. Simple, complex, and the derived lipids. Now let's talk about the phospholipids in somewhat more detail, starting with the glycerophospholipids, where I had said the glycerol is the alcohol, the glycerol is the alcohol, all right, where the esterified the acids are attached. The acid number 1, this is R1, the acid number 1 R1, the acid number 2 R2, and we said something else in this case is the phosphate which is attached, the phosphate which is attached, that is the attached at carbon 3. So this is the building block of the glycerophospholipids. The building block of glycerophospholipids. All the glycerophospholipids are further modified. They are further attached to an additional unit beyond the phosphate. They are further attached to an additional unit beyond the phosphate. So glycerophospholipids have a building block. This building block is known as the phosphatidic acid. This is the structure that is drawn in front of you. You need not draw the structure, but understand glycerol, to which the two fatty acids and one phosphate is attached. It is modified by addition of an additional group, which we'll see subsequently. Whatever additional group is attached, that is used for nomenclature. For example, if this additional group is, let's say, choline, then the name becomes phosphatidylcholine. If this is serine, then the name becomes phosphatidylserine. So like this, the name will change depending on which molecule is attached. The building block will remain the same. The phosphatidyl group is there, beyond which an additional group may be attached. Beyond which an additional group may be attached. So one very important glycerophospholipid is the phosphatidylcholines, to which choline is attached. To which choline is attached. The other name for the phosphatidylcholines is the lecithin. The other name for the phosphatidylcholines is the lecithin. So if you see the term lecithin in the question, immediately you should recall that we are talking about the phosphatidylcholine. We are talking about the phosphatidylcholine, where we will have the phosphatidic acid to which choline is attached. It is the most abundant phospholipid of the cell membrane. The most abundant phospholipid of the cell membrane. It is the phosphatidylcholine, and it represents a large part of the body store of the choline. It represents a large part of body store of the choline. Some of the phosphatidylcholine are very important. Some phosphatidylcholine are very important, particularly the dipalmitoyl lecithin. You know, dipalmitoyl lecithin is the surfactant. All of you know this. The dipalmitoyl acid is the surfactant. In some premature children, the production of the dipalmitoyl lecithin has not yet started, and these children will land up in the respiratory distress syndrome. Reason being, the dipalmitoyl lecithin is produced by the mature type 2 pneumocytes, and in the premature infant, they are not yet being produced. Okay. So dipalmitoyl lecithin is one of the very important phosphatidylcholine with a very specific clinical correlation. Deficiency in premature infants results in the respiratory distress syndrome. This question has been asked several times. You should remember which of the following phosphatidylcholine is deficient in RDS. The answer will be the dipalmitoyl lecithin. The dipalmitoyl lecithin. If we replace the choline by ethanolamine, then we get what is known as cephalin. The only important point to note here is, if I say cephalin, you should remember that cephalin will have the ethanol amine. Cephalin will have the ethanol amine. Okay, this is also present in the cell membrane. Nothing special about the cephalin. Only thing, remember, cephalin means we have the ethanolamine there in place of choline. Now we come to the phosphatidylserine. Now we come to the phosphatidylserine. This is very, very important. Obviously, here the serine will replace the choline. It is found in the cell membrane, but its orientation is different from what we see in case of the others. See, you remember that the cell membrane is a lipid bilayer. Cell membrane is a lipid bilayer. All right. So what happens normally? This is, let's say, outside, and this is the inside of the cell. All right, so normally the phosphatidylserine is present on the inner aspect of the cell membrane. Normally it is present on the inner aspect of the cell membrane, but when a cell is about to undergo apoptosis, at that time this phosphatidylserine will flip to the outside, and now it is projecting on the outside. Now it is projecting on the outside. So presence of phosphatidylserine on the outer aspect implies the cell is about to undergo apoptosis. All right, therefore it is the marker of apoptosis. Phosphatidylserine is the marker of apoptosis. So remember, phosphatidylserine normally present on the inner aspect. If it is present outside, means the cell is about to undergo apoptosis. Then we have what is known as phosphatidylinositol. Phosphatidylinositol is a very specific molecule. Here what is having the glycerol is attached to R1, it is attached to R2, and the phosphate is attached to inositol diphosphate. The phosphate is attached to inositol diphosphate. Now we have the enzymes called the phospholipases. The phospholipases, the different types are there, A, B, and C, which will cleave the attached groups at the different carbon 1, carbon 2, carbon 3. The phospholipase C will cleave the bond at carbon 3. It will cleave the bond at carbon 3. If the phospholipase C is acting here, what you get is IP3 and 2 fatty acid attached to glycerol. This is known as diacylglycerol. So action of phospholipase C on the phosphatidylinositol gives rise to two important molecules, which both of them are second messengers.
Papa, this is our feet and numbers. तो फिर ये
हाँ, बाद में अभी क्लास हो रहा है ना बेटा। अच्छा बोलो।
देखो ये फर्स्ट स्टेप और ये नंबर है,
ठीक है,
6 तक है 1 से।
ठीक है,
तो फिर ये फर्स्ट स्टेप भूल जाओ, नंबर्स को भूल जाओ
बेटा। लंबी स्टोरी है, बाद सुन ले। सारे लोग देख रहे हैं, इनको ही बताना पड़ेगा, इधर कैमरा के सामने आके बता दो। हाँ ठीक है, बता दो। अच्छा देख के जाना क्या।
अच्छा काम
ठीक है, हाँ क्या करेगा। I'm sorry, चलो। Okay, the phosphatidylinositol I told you is acted upon by the phospholipase C, and it gives rise to the very important second messengers, the inositol diphosphate, inosine diphosphate, and sorry, inositol triphosphate, IP3, and the diacylglycerol. Both of them work as second messengers. So this is a very important role of the phosphatidylinositol. It is the precursor of the second messengers. It is the precursor of the second messengers. Okay. So, like I said, precursor of second messengers, the IP3 and the diacylglycerol generated from the phosphatidylinositol. Then two very important molecules which at first look may not appear to be the glycerophospholipids. First is the cardiolipin. First is the cardiolipin. Cardiolipin, if you look, is made up of two phosphatidic acid attached to the glycerol. So can anyone tell me how many glycerol are there in cardiolipin? How many glycerol are there in cardiolipin? This question you should be able to answer because the structure is in front of you. I've already told you what we mean by the phosphatidic acid. Therefore, it should be a straightforward question for all of you. How many glycerol are there in cardiolipin? Anyone? How many glycerol in cardiolipin? Your options are 1, 2, 3, 4. Very good, very good. So please note one glycerol in the phosphatidic acid, another glycerol in phosphatidic acid, and third glycerol through which the two phosphatidic acid are linked. So remember the number of glycerol in cardiolipin is 3. First point. The number of glycerol in cardiolipin is 3. Okay. And now the cardiolipin is present in the muscles. The cardiolipin is present in particularly in the muscles because it is a component of the mitochondria. It is found only in the mitochondria and is important for the mitochondrial function. It is very important for the mitochondrial function. So it is critically important for both the muscles, whether it is the skeletal or the cardiac muscle. And deficiency gives rise to what is known as the cardioskeletal myopathy. Deficiency gives rise to what is known as the cardioskeletal myopathy. The cardiac muscle, the skeletal muscle, both are grossly affected when cardiolipin deficiency is there. In cardiolipin deficiency, both are grossly affected. And then we have the lysophospholipids. Now, the lysophospholipids, strictly speaking, are not the glycerophospholipids. They are intermediates. They are strictly speaking not the glycerophospholipids, but they are intermediates in the metabolism of the phosphoglycerols. Here, we only have a single fatty acid. What it means is something like this. See, we have the glycerol, where normally the fatty acid 1, fatty acid 2, phosphate and choline are attached, right? Now these R1 and R2 are not fixed, they are continuously getting recycled. The R1 will go out, the R1 will go out, and in place of R1 now maybe R3 will come in, R3 will come in. Okay, now imagine the situation when R1 has gone out and R3 has not yet come in. This means that this place is empty. This place is empty. This place is empty. So this is what we call the lysophospholipid. This is what we call as the lysophospholipid. Here in place of that we will have the hydroxyl group. So this is the lysolecithin, where only one fatty acid is present. Now Why is this structure important? Why is the lysolecithin important? The lysolecithin is important because this particular group which is present is very reactive. Because it is reactive, it tends to get oxidized. If it gets oxidized, it is taken up by the macrophages. Macrophages are unable to keep track of the oxidized lipids that they consume, so they'll keep taking the oxidized lipids, and gradually they will become very large when we call them the foam cells. And all of you know that foam cells are implicated in the causation of atherosclerosis. Foam cells are implicated in the causation of atherosclerosis. That is why we are interested in the lysophospholipids. They are strongly associated with the incidence of atherosclerosis. They are strongly associated with the incidence of atherosclerosis. Okay. So remember, strictly speaking, these are not the glycerophospholipids. They are an intermediate in metabolism of the glycerophospholipids. We have another molecule which is strictly speaking not a glycerophospholipid, about which you should know, and that molecule are the plasmalogens. Plasmalogens are molecules similar to the glycerophospholipids, but there is one very fundamental difference. What is that difference? Let's see. Here, in place of an alcohol, sorry, uh, in place of the ester link, we have an ether link. What do you mean by ether link? See, in the glycerol, where we have the hydroxyl group, here the carboxylic group will come and attach. When this attaches, what we get is an ester bond. What we get is an ester bond. This is the ester bond. But in plasmalogen, instead of the carboxylic group, what we have here is another alcohol. What we have here is another alcohol. So when the bond formation occurs, the bond formation is something like this. This is known as ether bond. So one of the fatty acid is replaced by an alcohol. One of the fatty acid is replaced by an alcohol. Typically it is an unsaturated alcohol. Typically it is an unsaturated alcohol. Okay, so these are called the plasmalogens. These are known as the plasmalogens. Plasmalogen, as you can see, will occur in the brain and muscle. They are very important for the neural development. They participate in the cell-cell interaction. They will participate in the cell-cell interaction, which is a very important role for a lipid. Okay. So these are the important glycerophospholipids and their derived molecules. Next, we go to the second group, that is the sphingophospholipids. First, we talked about the glycerophospholipids, where we said we have the building block called the phosphatidic acid, in front of which the other molecules can be attached, based on which we have the different names. We briefly had a look at some of the important glycerophospholipids. Now we are moving on to what is known as the sphingophospholipid. In the sphingophospholipid also we have a building block. This building block is what is known as ceramide. In ceramide we have two things: the alcohol, which is sphingosine, and to the alcohol the fatty acid is attached. This is called the ceramide, the building block of sphingolipids, not only phospholipids but the sphingoglycolipids also. In sphingoglycolipids also the ceramide will be the building block. In front of this molecule, in front of this molecule, the other groups can be attached. Other groups can be attached. For example, we are talking about the sphingophospholipid, so the phosphate will get attached here, and to the phosphate there will be attachment of the choline. In sphingophospholipid, we only have the subgroup where choline is present. The only subgroup we have is the where choline is present, and it is known as the sphingomyelin. So we have only one subgroup in the sphingophospholipid. In the sphingoglycolipids, we will have multiple subgroups. In the sphingophospholipid, we have only one subgroup, which is called the sphingomyelin. It is found in large quantities in brain and in the nerve tissue. And if you look at the structure, it will give you the fatty acid, only one fatty acid, only one fatty acid, whereas in the glycerophospholipids we had R1 and R2, two fatty acid were there. The phosphate, which is released as the phosphoric acid, I showed you the choline is there, and obviously the alcohol is sphingosine. The alcohol sphingosine attached to one fatty acid, subsequently attached to a phosphate and a choline. So every time you cleave the sphingomyelin, this is what you are going to get, unlike the glycerophospholipid where we get two fatty acid, we get R1 and R2. Remember, in sphingomyelin, the glycerol is not present. Obviously, the alcohol is sphingosine, so there is no question of the glycerol being present. It is the sphingosine which is the alcohol. So this is the structure. You don't need to remember the structure. I'm just showing you the structure of the sphingomyelin, where to ceramide first the phosphoric acid is attached, to which the choline is attached. Now let's talk about the glycolipids. The glycolipids, remember, are having the alcohol sphingosine. All glycolipids have the alcohol sphingosine, so they are also known as the glycosphingolipids. So they are also known as the glycosphingolipids. Okay. So what is the structure of the glycosphingolipids? The lipid containing fatty acids, sphingosine, and a carbohydrate. The carbohydrate can be a monosaccharide, it can be oligosaccharide, it can be something which has oligosaccharide in addition to the sialic acid. So different type of carbohydrate structures may be attached. They are very, very important for the CNS. Very, very important for the CNS. They occur in the outer leaflet. Again, they are important in cell-cell interaction. So they occur in the outer leaflet. All right, and the different structures of the carbohydrate are present, based on which the glycolipids are further subdivided. Based on the carbohydrate structure, they are further subdivided. The subgroups are cerebroside, sulfatide, globoside, and ganglioside. We'll look at them one by one. In cerebroside, we have the ceramide, we have the ceramide plus a monosaccharide. In sulfatide, we have ceramide plus a sulfate plus a monosaccharide. In ceramide, we have attachment of sulfate and monosaccharide to give us the sulfatide. In globoside, we don't have the monosaccharide, rather we have the oligosaccharide. And in ganglioside, we have the ceramide plus oligosaccharide plus, as mentioned, the sialic acid. As mentioned, the sialic acid. Okay. So one or more sialic acid may be present. One or more sialic acid may be present. Let's look at these glycolipids very, very quickly. Cerebroside, I told you, ceramide plus a monosaccharide. Depending on which monosaccharide is there, either it can be the galactose or it can be the glucose. The galactosyl ceramide are mainly present in the CNS. That is why galactose is a very important molecule for the growing infant. It is required for the development of the brain. It contains the galactose. The milk which contains the lactose, which gives us galactose, is very, very important for the development of the brain. The galactosylceramide will require the galactose. The glucosylceramide is mostly present in the extraneural tissue. It is mostly present in the extraneural tissue. So galactosylceramide and the glucosylceramide, galactosylceramide and the glucosylceramide. Sulfatide, I already told you, the ceramide and a sulfated monosaccharide, they are present in the myelin. The globoside are ceramide plus an oligosaccharide. Last we come to the ganglioside. Ganglioside we said ceramide, oligosaccharide with at least one sialic acid residue. Depending on the number of sialic acid residues, they are further subdivided into GM, GD, GT and GQ. Depending on number of sialic acid residues, they are further subdivided into GM, GD, GT and GQ. Okay, so please note, till now we have talked about the alcohol, we have talked about the presence of carbohydrate, we have talked about the presence of sialic acid, but we have not talked about the size of, we have not talked about the size of the uh, carbohydrate chain. So depending on the size of the carbohydrate chain, each of these subgroups can be further subdivided. For example, if you do the electrophoresis, you get the GM1, GM2, GM3, depending on the size of the carbohydrate chain. Depending on the size of carbohydrate chain, that they will move to a different distance and they can be further subdivided. This separation can be identified with the help of electrophoresis. Which is among the GM1, GM2, GM3, which is the simplest ganglioside? Which is the simplest ganglioside? Is it GM1, is it GM2 or GM3? The simplest ganglioside is GM1, GM2 or GM3? Which is the simplest ganglioside? GM1, GM2 or GM3? The answer is not GM1, the answer is GM3. The simplest ganglioside is GM3. Please note, the simplest ganglioside is GM3. However, GM1 is clinically very important. GM1 is clinically very important because it acts as the receptor for cholera toxin. It acts as a receptor for cholera toxin. Very good. GM3. GM1 will act as receptor for the cholera toxin. So these are some of the very important points related to the glycolipids that you must remember. The classification, what are the groups present, and what is the simplest and what is the most important ganglioside. They are respectively GM3 and GM1. GM1 is the receptor for cholera toxin, and it will cause the cholera in response to the toxin attaching to the receptor. Now, these sphingolipids, the phospholipids, the sphingophospholipids, as well as the sphingoglycolipids, all of them can have defective degradation. Because of the defective degradation, they can start accumulating in the body, and as a result, give rise to what is known as sphingolipidosis. I'll repeat here: the sphingolipidosis can result from accumulation of the sphingoglycolipids as well as the sphingophospholipids. So sphingolipid contains both types, the sphingophospholipid and the sphingoglycolipids. It is a very important inborn error of metabolism due to defective metabolism of the sphingolipids. Just like we had the mucopolysaccharidosis, they were being synthesized but not being degraded. Similarly, the sphingolipids are being synthesized but not being degraded. As a result, they will start accumulating. Let's see. The defect is in catabolism. The defect is in catabolism of lipids containing sphingosine. It is also a subtype of lysosomal storage disorder. It is also a subtype of lysosomal storage disorder. I'll give you the names of the sphingolipidosis. Some common features to remember are all of them are autosomal recessive. All of them are autosomal recessive. All of them will present with cherry red spot. All of them will present with cherry red spot. All of them will present with mental retardation. These are three very important points to remember about sphingolipidosis. All of them are autosomal recessive. All of them will present with cherry red spot. All of them will present with mental retardation. However, there are certain exceptions. The Fabry's is X-linked recessive. The Fabry's is X-linked recessive. Cherry red spot is not seen in Fabry's and Gauchers. Cherry red spot is not seen in Fabry's and Gauchers. Mental retardation is also not seen in Fabry's and Gauchers type 1. There are three types of Gauchers, type 1, 2 and 3. In type 1, the mental retardation may not be seen. Very, very important points. The zebra body inclusion in Niemann-Pick. These zebra body are visible via electron microscopy. They are visible via electron microscopy. And then we have the globoid cell inclusion in crabbies. We also have what is known as crumpled or wrinkled tissue paper appearance. Cells having crumpled or wrinkled tissue paper appearance. Alright, these are known as the Gaucher cell. These are known as the Gaucher cells. So these are some of the very important findings to know. Remember, Gaucher cell are characteristic but not diagnostic of the Gaucher's disease. They are characteristic but not diagnostic of the Gaucher disease. The globoid cell, obviously they are not very clear. If you try to visualize, globe like inclusions are there in the cell. Here also globe like inclusions present in the cell. These are the globoid cell in Krabbe's disease. As you can see, they are visible under light microscope. They are visible under the light microscope. The blue tubes are highlighting the globoid cells. And then we have what is known as the zebra body inclusion. Remember, I told you it is seen only under the electron microscopy. It is seen only under the electron microscopy. It comprises of the concentric membrane bound lysosomal inclusions. Various approaches used for treatment of the sphingolipidosis. Some of them are enzyme replacement therapy. I told you once we complete all the inborn errors, then I'll show you the list of enzyme replacement therapies. Bone marrow transplant. Bone marrow transplant is supposedly one of the quite effective treatment modalities, but the problem is finding a matched donor for the bone marrow transplantation. Finding a matched donor for the bone marrow transplantation is the biggest challenge. If it is possible, then these two together can be very effectively used for management of the disorder. Both of them are available for treatment of Gauchers and Fabry's. Both of them are available for treatment of Gauchers and the Fabry's. Some other approaches which can be used are substrate deprivation and the chemical chaperone therapy. Substrate deprivation and chemical chaperone therapy. They are still in the experimental stages. They are not available clinically. They are not available clinically. What is available clinically is the gene therapy, and this is supposedly the best possible answer to a single enzyme defect. Single enzyme defect can be very correctly remedied by the gene therapy. Now, if you talk about gene therapy, it can be of two types. Previously, it was the in vitro type. Previously, it was the in vitro type. The in vitro type has a very low success rate. Very low success rate. Then came what is known as ex vivo. Now you'll note we normally say in vivo. In vivo means inside the body. Ex vivo means you're going outside the body, but everything is occurring inside the biological cell. We are not doing it in a chemical system. We are doing it inside the biological cell. This is called the ex vivo gene therapy. Ex vivo gene therapy has a much higher success rate after a single session. Approximately 80% success rate is there. Obviously, it is much more costly. It is much more costly. So in vitro gene therapy, what we are doing, we take a vector, we select a vector, and to this vector we add the corrected gene. We add the correct gene copy, and then we infuse into the patient, and then we infuse this into the patient, and in now only thing we can do is wait for the uptake of the vector into the target tissue. Because of not being able to target the tissue correctly, and not being able to target the you can say stem cells. See, if the vector is taken up by the somatic cell, after certain cell divisions it will die. So even if that uptake is successful, it will not be prolonged. So what we do in the ex vivo system, we try to minimize the factors which decreases the effectiveness. What are the factors? Identify or reach target tissue, and second, identify transfuse stem cells. So we try to minimize both the variables in the lab itself when we are using the ex vivo gene therapy. So what we do there, we extract stem cells directly. We are directly extracting the stem cells by the technique called the flow cytometry. Remember, the flow cytometry can be used for just the identification if you're using the antibodies, or if you're using the antibody tagged with magnetic beads, they can also be used for separation. So by using antibody with magnetic beads, we use the flow cytometry to extract the stem cell. So identifying the stem cell work is done. So now once you've extracted the stem cell, we introduce the vector by one of the various mechanisms which are used. Not the vector, we add the corrected gene segment. Various modalities are in use to add this gene segment. Various modalities are in use to add this gene segment. For example, we can do the traditional vector approach. We can use the traditional vector approach. We can use what is known as electroporation, or we can use what is the newer modality which has much higher success rate. It is the gene gun. However, a lot of experience and expertise is required in handling these gene transport mechanisms. Okay, so these are the various possible approaches to treatment. I've told you a little bit about the gene therapy because it is one of the upcoming techniques of management, and it is likely to be asked in your exam. You should know about the in vitro and the ex vivo gene therapy. In vitro, we are only using the vector and the corrected gene, and we are just directly infusing into the patient. Okay, so vector corrected gene are working outside. Rather, we should call this one also in vivo and ex vivo. Okay, so this was about the possible approaches to treatment, and this is the list of the sphingolipidosis. the list of the sphingolipidosis and the corresponding enzyme deficiencies. I request you to kindly note down this list. Most of your guides and books may not mention the Sandhoff disease, but remember, Sandhoff disease is also a type of sphingolipidosis. Okay, so the various sphingolipidosis, the Tay-Sachs disease, where the hexosaminidase A is absent, the Sandhoff disease, where both hexosaminidase A and B are deficient. Then we have the Fabry's disease. Remember, Fabry's disease was the X-linked recessive disorder, where alpha galactosidase is not there. We have the metachromatic leukodystrophy. The aryl sulfatase is missing. In which disorder we find the deficiency of aryl sulfatase B? Then we have the Krabbe's disease. In Krabbe's disease, the beta galactosidase is deficient. We have the Gaucher's disease, where beta glucosidase is deficient. The Niemann-Pick, where deficiency of sphingomyelin is there, and the Farber's, where deficiency of ceramidase is there. Okay. Now, one very important question. If you recall, I had told you in sphingolipidosis we have the accumulation of the uh, sphingophospholipids as well as the sphingoglycolipids. All right. So you have to tell me in which all conditions the sphingophospholipid will accumulate. In which all conditions the sphingophospholipid will accumulate. I'll number them so you can write the number directly in the answer. Very good. Mucopolysaccharidosis type 6 Maroteulami was the disorder where we use deficiency of aryl sulfatase B is seen. I am numbering the disorders which you can use to write in the answer. In which of the following conditions the sphingomyelin or the sphingophospholipid accumulation will occur? I have given you the hint. The accumulation of the sphingophospholipid occurs in which of these conditions? I have written the number. You can use the numbers to write your answer. In which all conditions the sphingophospholipid will accumulate in the body? The sphingophospholipid, also known as the sphingomyelin. That's your hint. Send in your answers fast. Send your answers fast. Please note, among the whole list, the sphingomyelin accumulates only in one condition, and that is the Niemann-Pick disease. In all other disorders, the accumulation of sphingoglycolipid is occurring. If you look at the enzymes, you will realize all of them are acting on the carbohydrate part. All of them are acting on the carbohydrate part. So obviously, there the accumulation of the sphingoglycolipid is going to occur. The Niemann-Pick, as correctly mentioned by several of you, is the only disorder where the sphingomyelin accumulation occurs. It is the only condition where sphingomyelin or the sphingophospholipid accumulation occurs. In all others, it is the sphingoglycolipid which is going to accumulate, whether it is the Tay-Sachs, Sandhoff, Fabry's, metachromatic, Krabbe's, Gaucher's, or the Farber's. Okay, so this is a very important distinction. You should remember sphingomyelin accumulates only in the Niemann-Pick. In all other, the sphingoglycolipid is going to accumulate. Second question, which of the following disorders is the most common? Which of the following disorders is the most common? Most common among all the disorders. Which is the most common among all the sphingolipidosis? Quickly tell me the answer. The most common among all the sphingolipidosis. I'm waiting for the answers. I'm still waiting for the answers. The correct answer, the correct answer is Gauchers. The Gauchers is the most common sphingolipidosis. Gauchers is the most common sphingolipidosis. So let's take a brief look at Gauchers. A very brief look at the Gauchers. Gaucher's disease, like I told you, is the most common sphingolipidosis. Not only sphingolipidosis, it is the most common lysosomal storage disorder. Very good. The correct answer is Gaucher's. It is the most common lysosomal storage disorder. Defective enzyme is the beta glucosidase, also known as glucocerebrosidase, or the glucosylceramidase, also known as glucocerebrosidase or the glucosylceramidase. We already said the cherry red spot and the mental retardation are not seen. Gaucher cell, which is seen here, has the appearance of wrinkled or crumpled tissue paper appearance. Wrinkled or crumpled tissue paper appearance. Typical presentation in these patients is with acute abdominal pain and the bone pain. Acute abdominal pain and the bone pain. Two terms were associated with type 2 Gauchers. No need to go into details, just remember the terms: collodion babies and arthrogryposis. Collodion babies and arthrogryposis, they are two terms associated with the Gaucher's disease type 2. What is more important is the simultaneous elevation of the chitotriosidase, the ACE enzyme, and TRAP enzyme. The chitotriosidase, ACE, and TRAP simultaneously being elevated has a very strong indication that the condition is the Gaucher's disease. Recently, to this list, we have added the CCL18 to increase the sensitivity. To increase the sensitivity, we have added the CCL18, also known as the PARC. So if all of these are simultaneously elevated, it is a very strong indication that we are dealing with the Gaucher's disease. Remember, the confirmation will only be via the genetic analysis. The confirmation will only be via the genetic analysis. Okay, so this is a little bit about the different sphingolipidosis, particularly about the Gaucher's disease. If you have any questions so far, please let me know. Else we move on with the discussion in the lipids. No questions? If there are no questions, then I'll go ahead. I'll go ahead. We'll talk now about one of the blocks of building units of the lipids, that is the fatty acid. We had seen in the lipids, we have the fatty acid, we have the alcohol. So I'll talk a little bit about the fatty acid which are present inside the lipids. Now some questions are coming from the fatty acid and their classification also. And if you're clear about the classification, it will also help you in the pharma and the medicine section, where we have the icosanoids coming in, the thromboxane, the prostaglandins. All of them are coming from the fatty acids only. So let's take a look. The fatty acids are named after the corresponding hydrocarbons. So mainly they will occur as the natural fats and oils. And in our body, mostly we have the fatty acids with even number of carbon atoms. The odd chain fatty acid in our body is very, very rare. The odd chain fatty acid is extremely rare. Please note, fatty acid is never free. When we call it the free fatty acid, it is actually attached to the albumin. When it enters the cell, it is attached to what is known as the fatty acid binding protein, and the shorter chains uh, may exist as an ionized form. So the fatty, if you call it free fatty acid, mostly we should talk about the, we can say, small chain fatty acid. The correct term when we talk about the free fatty acid should be non-esterified fatty acid. So if you see this term NEFA, don't get worried. NEFA stands for non-esterified fatty acid. It is the technically correct term for the free fatty acid. It is a technically correct term for the free fatty acid. The non-esterified fatty acid should be more commonly used rather than the free fatty acid. We classify the fatty acids into two broad groups: saturated and unsaturated. Two broad groups: saturated and the unsaturated. Within the unsaturated, we have another group known as the essential fatty acid. This is a subtype of the unsaturated fatty acids. The subtype of the unsaturated fatty acids. Now, when we talk about the saturated fatty acids, we divide them into further groups: the small chain, the medium chain, the long chain fatty acid, and we also have something known as the very long chain fatty acid. So the saturated fatty acid are further subdivided into the small chain, medium chain, long chain, and the very long chain fatty acid. The very long chain fatty acid are typically in the range of 20, typically in the range of 20 or more. We call them the very long chain fatty acid. This is for the saturated fatty acids. For the unsaturated fatty acids, the same classification can apply, but we have a better system to identify them, and this is what we have. The unsaturated fatty acid primarily classified as monounsaturated and the polyunsaturated. The polyunsaturated fatty acids give rise to what is known as icosanoids. Icosanoids are typically having 20 or more carbons. 20 or more carbons. The icosanoids are further subdivided into lipoxins and the leukotrienes. They are further subdivided into lipoxins and the leukotrienes, along with the prostanoids. Prostanoids include the prostaglandin, the prostacyclin, and the thromboxanes. The prostanoids include the prostaglandin, prostacyclin, and the thromboxane. Talking about the various types of fatty acid, very commonly we get this question: which fatty acid is more cardioprotective, which fatty acid is less cardioprotective? So remember, the most cardioprotective fatty acid as per current literature is the omega 3 PUFA. Omega 3 PUFA is the most cardioprotective fatty acid, followed by the MUFA. So as per current literature, most cardioprotective fatty acid is the omega 3 PUFA. I told you some of the pupa are known as the essential fatty acid. Some of the pupa are known as essential fatty acid. The most important being the linoleic acid. Linoleic acid, I will show you a chart where the sources are described. No need to write. The humans cannot synthesize the omega 3 pupa. In addition, they also cannot synthesize the omega 6 pupa. They cannot synthesize omega 3 pupa and the omega 6 pupa. Anything beyond omega 6 can be synthesized. 6 and below cannot be synthesized by the body, and therefore they'll become the essential fatty acid. Okay. Some of the essential fatty acids can act as precursors, as written here. The alpha lanolinic acid can give rise to the EPA and the DHA. It can give rise to the EPA, eicosapentaenoic acid, and docosahexaenoic acid. Docosahexaenoic acid. To summarize, where is that list? You don't have that list. Okay. To summarize, we have two broad series, the omega 3 and the omega 6 series, which cannot be synthesized in the body. The omega 3 series and the omega 6 series, both of them are the PUFA. The linoleic acid belongs to omega 6 series. Linoleic acid belongs to omega 6 series. The alpha linolenic belongs to the omega 3 series. The alpha linolenic belongs to the omega 3 series. Now, as said, the alpha linolenic acid can be converted to eicosapentaenoic acid or the docosahexaenoic acid. The linoleic acid can be converted to gamma linolenic acid, and this gamma linolenic acid can then be converted into the arachidonic acid. So remember which are the omega 3 fatty acids and which are the omega 6 fatty acid. This question is very frequently asked. All of the following are omega 3 except, all the following are omega 6 except, which of the following is omega 3, which of the following is not an omega 3. So this question is asked repeatedly again and again. Therefore you should remember this list. Which of the fatty acids are omega 3, which are the fatty acids are omega 6. Now this is again very important. This comes again and again repeatedly, and the options keep shuffling from one side to another, and every time they change the option, the correct answer will also change. All right. So dietary sources of linoleic acid, this is by far the most important fatty acid that you are going to mark. See, I've shown you here that you are supposed to, what happened? Okay. Among these, if you have to mark one answer, the most important essential fatty acid will be the linoleic acid. Most important, the PUFA will be linoleic acid. But if you are able to mark two, remember linoleic and the alpha linolenic, both are required to synthesize the full spectrum of the essential fatty acid. So until unless you have both, you cannot synthesize all of them. So linoleic plus the alpha linolenic acid. Now because we have the gamma linolenic acid which can be obtained from linoleic, if you have to choose one, the linoleic acid becomes the better answer. But if the option says linoleic acid and the alpha linolenic acid, obviously that is the best correct answer. So here we come. to the dietary sources of the linoleic acid. The dietary sources of the linoleic acid, richest source is the safflower oil. A richest source is the safflower oil, followed by the corn oil, and then we have the sunflower oil. Safflower oil, corn oil, sunflower oil are very very important. Lastly, we have what is called the soybean oil. You can see different percentages. Corn oil, sunflower oil are quite close in terms of content. They are quite close. I also give you the dietary source for all the other essential fatty acids. Dietary source for all the other essential fatty acids. For linoleic, as you can see, the richest source is safflower oil. For linolenic acid, it is the soybean oil. Arachidonic acid coming from the animal products, the meat, egg, and milk. The EPA coming from fish oil. The DHA coming from the breast milk. The DHA coming from the breast milk. Okay. Here I'll ask you to take a very very short break of let's say 10 minutes. You should refresh yourself because now we are going to go into the oxidation of fatty acid, and it is a slightly conceptual topic. So you should refresh yourself, have a cup of tea, and then we'll quickly do the oxidation of fatty acids. This 9:15. We'll restart at the 9:25. Quickly restart 9:25. Have a cup of tea, and then we go into the details of the oxidation of fatty acids. Very very short break, 10 minutes, just for a cup of tea.